Welcome to Sleepy Science Facts, a place where learning drifts softly into the quiet of the night. Tonight, we wander into the secret life of plants, a calm, tender journey into the hidden world beneath every leaf, every root, every fragile petal. As the world slows down around you and your breathing settles into its own peaceful rhythm, let these gentle truths wrap around you like a warm blanket, guiding you into a soft state of wonder. If these soothing science stories bring you comfort, I'd be truly grateful for your support. A like, a subscribe, or sharing this with someone who loves peaceful knowledge helps this little channel grow quietly, steadily, just like the plants we're learning about tonight. Now, take one slow breath and let's slip into the quiet green world together to uncover the secret life of plants. Plants are the quiet, unseen heroes of our world. They stand rooted in the soil, facing the sun, wind, and rain, yet they never complain. From the tiniest moss on a rock to the tallest tree touching the clouds, plants are everywhere, bringing life, beauty, and balance to our planet. They are the green lungs of the earth, breathing out oxygen and giving us the air we need to survive. Without them, life as we know it would simply not exist. Plants are more than just greenery. They are living, feeling organisms that respond to their surroundings. They sense light, temperature, water, and touch. They grow toward the sun, stretch their roots into the earth to drink, and even communicate with each other through subtle chemical signals. Each leaf, stem, and root tells a story of patience, resilience, and quiet strength. Watching a plant grow reminds us that life is a delicate balance, shaped by time, care, and the environment. One of the most magical things about plants is their ability to transform energy. Through a process called photosynthesis, they capture sunlight and turn it into food, fueling their growth and sustaining other forms of life. In doing so, they provide nourishment for animals, humans, and countless microorganisms. Plants are, in essence, life's first chefs, creating energy from sunlight and water and sharing it generously with the world. Plants also teach us the power of adaptation. In deserts, cacti store water in their thick stems. In rainforests, towering trees reach for light above a crowded canopy. In cold mountains, small alpine plants cling to rocks, surviving freezing winds. No matter how harsh the environment, plants find a way to live, showing us the strength of persistence and the beauty of resilience. Their lives are a lesson in patience, reminding us that growth is often slow but steady, and survival comes from learning to adjust to challenges. Beyond their practical importance, plants touch our hearts in countless ways. They offer shade on hot days, fragrance in the morning air, and colorful blooms that lift spirits. Gardens, forests, and fields create spaces of calm, joy, and inspiration. Even simply watching a seed sprout or a flower bloom can bring a sense of wonder and hope. Plants connect us to nature and remind us of life's simple pleasures. In every corner of the earth, plants form the foundation of ecosystems. They provide food, shelter, and protection for animals. They stabilize soil, purify air, and regulate water cycles. Their roots bind the land, their leaves catch rain, and their presence shapes the climate. Life in all its diversity depends on plants. Ultimately, plants are silent teachers, generous providers, and resilient survivors. They demonstrate patience, adaptation, and interconnectedness. By caring for plants and understanding their lives, we deepen our connection to nature and ourselves. Plants are not just living beings. They are a reminder that life thrives in quiet strength, gentle persistence, and unwavering beauty. Plant photosynthetic acclimation to extreme heat in the most sun-drenched and unforgiving regions of the world. where the earth cracks under relentless heat and the air shimmers with mirages, certain plants thrive in conditions that would wither most life. In these harsh, breathless landscapes, every ray of sunlight feels heavy, yet some life forms rise with quiet strength. Among these extraordinary survivors is Tidstromia oblongifolia, a shrubby desert species that flourishes in the heat of Death Valley, where summer temperatures routinely soar beyond 45 degrees Celsius. Observing this plant in its natural habitat is like witnessing a masterclass in survival. Its leaves glint faintly in the sun, carrying a subtle, almost defiant glow, not merely enduring the heat, but performing the delicate work of photosynthesis with remarkable efficiency. This plant has evolved strategies that allow it to maintain growth, capture sunlight, and conserve water, all under temperatures that would cripple most other species. Its quiet resilience feels almost emotional, a reminder that even in the world's harshest corners, life refuses to give up. What makes Tidstromia particularly fascinating is its ability to adjust the very machinery of photosynthesis in response to extreme heat. Photosynthesis, the process by which plants convert sunlight into chemical energy, typically has an optimum temperature range. Beyond that range, enzymes and proteins begin to falter, and energy production plummets. Yet Tidstromia has been observed to shift this optimum rapidly, within days, allowing it to thrive even when exposed to intense heat. Laboratory studies simulating Death Valley conditions have shown that within just 2 days of heat exposure, this plant can recalibrate its photosynthetic systems, enabling it to maintain carbon assimilation rates far above what would be expected under such conditions. Over a span of 10 days, biomass accumulation can triple compared to cooler conditions, illustrating an extraordinary resilience that surpasses nearly every common crop plant. This ability to not just survive but flourish under extreme conditions is a testament to the plant's remarkable physiological plasticity, a quiet triumph written into its cells. At the cellular level, the plant employs an intricate strategy to safeguard its photosynthetic machinery. Chloroplasts, the tiny green organelles responsible for converting light into chemical energy, undergo a remarkable transformation in shape, taking on a distinct cup-like configuration. This shape is believed to enhance the internal diffusion of carbon dioxide and optimize the absorption and processing of sunlight, distributing energy efficiently across the leaf tissues. Meanwhile, mitochondria, the cell's energy-producing powerhouses, move closer to chloroplasts during periods of extreme heat. This proximity allows mitochondria to absorb excess, reducing power and reactive molecules generated by chloroplasts, effectively preventing cellular damage. By coordinating energy production and consumption in this way, the plant maintains photosynthetic function and protects itself against the accumulation of harmful reactive oxygen species, which can destroy proteins, lipids, and DNA. This delicate harmony between organelles feels almost like a silent conversation, a collaboration forged by the pressures of the desert. The secret to this incredible resilience is found in the plant's genetic program. Exposure to heat triggers a massive reorganization of gene expression, activating thousands of genes that produce proteins responsible for protecting and repairing the photosynthetic machinery. These include chaperone proteins that stabilize delicate enzyme complexes, antioxidant enzymes that neutralize harmful molecules, and structural proteins that maintain the integrity of organelles. What is particularly striking is the speed of this genetic response. Within days, the plant is able to adjust its physiology and metabolism to cope with temperatures that would be lethal to most other species. This dynamic genetic flexibility is a key component of its survival strategy, allowing it to adapt to the unpredictable and extreme environment of the desert. It is evolution in motion, swift, purposeful, and profound. From an ecological perspective, the adaptations of Tidestromia are not only impressive, but also essential. In the desert, water is scarce, temperatures fluctuate dramatically, and sunlight can be overwhelming. By adjusting its photosynthesis, optimizing energy use at the cellular level, and protecting itself from damage, the plant ensures that it can grow, reproduce, and complete its life cycle in a habitat where survival is precarious. Its leaves act as finely tuned solar panels, capturing energy while regulating temperature and conserving precious moisture. This combination of physiological, cellular, and genetic adaptations exemplifies the remarkable ingenuity of evolution in shaping life to endure extreme conditions, a quiet brilliance hidden beneath sunlit leaves. For scientists, studying Tidestromia and similar thermophilic plants provides invaluable insights into how life can adapt to extreme heat. These findings are particularly relevant in the context of global climate change, which is increasing the frequency and intensity of heat waves around the world. Many crop plants are highly sensitive to elevated temperatures, with yields declining rapidly as the environment pushes beyond their optimal range. By understanding the mechanisms that allow desert plants to thrive under extreme heat, researchers hope to identify strategies for engineering or breeding crops that can better withstand high temperatures, ensuring food security in a warming world. There is a quiet hope in this research, a belief that the wisdom of desert plants may guide us toward a more resilient future. In practical terms, the lessons from Tidestromia could guide the development of heat-resilient crops. Traits such as chloroplast morphological plasticity, mitochondrial repositioning, and rapid gene expression reprogramming could be targeted in breeding programs or through biotechnological approaches. For example, introducing or enhancing genes that control organelle mobility or stress-responsive proteins in crops might enable them to maintain photosynthesis under extreme heat. Moreover, these strategies could be combined with conventional breeding techniques that select for drought tolerance, water use efficiency, and other stress-resilient traits, creating plants that are truly equipped to survive in challenging climates. It is a vision of agriculture shaped by nature's toughest teachers. The study of Tidestromia also challenges our assumptions about the limits of plant life. It demonstrates that survival under extreme heat is not merely a matter of endurance, but of active adaptation. The plant's ability to dynamically adjust its physiology and metabolism highlights the complexity and resilience of natural systems. As researchers continue to unravel the genetic and biochemical mechanisms underlying these adaptations, they reveal a blueprint for life that is both elegant and powerful, offering hope for sustainable agriculture and a deeper understanding of the natural world. There is something profoundly moving in this, the idea that even in silence, evolution crafts solutions of astonishing beauty. Ultimately, the story of Tidestromia oblongifolia is a story of survival, adaptation, and ingenuity. It reminds us that even in the harshest environments, life finds a way to thrive. The plant's leaves, cells, and genes work in harmony to capture sunlight, protect against heat, and maintain growth, providing a window into the extraordinary capabilities of nature. As we face a warming planet, the lessons from this desert survivor could help guide us toward crops and ecosystems capable of enduring the challenges ahead, ensuring that life continues to flourish even under the sun's most intense gaze. It is a reminder, gentle yet powerful, that resilience is written into the very fabric of life. Mitochondria chloroplast interactions under stress inside every plant cell. An intricate ballet unfolds, a hidden choreography that allows life to flourish even under the harshest conditions. At the center of this dance are two vital organelles, the chloroplast and the mitochondrion. Chloroplasts, the green engines of photosynthesis, capture sunlight and transform it into chemical energy, fueling the growth and survival of the plant. Mitochondria, the cell's powerhouses, take this energy and convert it into a form that can be used for countless cellular processes. Under normal conditions, these organelles work in harmony, but under environmental stress, such as extreme heat, drought, or high-intensity sunlight, their relationship becomes even more critical, evolving into a tightly coordinated partnership that safeguards the plant's survival. It is nothing short of awe-inspiring, an emotional symphony of cellular cooperation that keeps life alive against all odds. Scientists have discovered that under stress, chloroplasts and mitochondria do more than just coexist. They actively interact. Advanced imaging techniques, including high-resolution confocal microscopy, have revealed that mitochondria often migrate closer to chloroplasts, forming transient physical contacts that facilitate the direct exchange of energy, metabolites, and even signaling molecules. These interactions allow the plant to balance energy production, prevent damage, and maintain photosynthetic efficiency when external conditions are hostile. It is a delicate but vital strategy. Chloroplasts generate energy in the form of ATP and reducing molecules, but excessive energy can create harmful reactive oxygen species, or ROS, which can damage proteins, membranes, and DNA. By positioning themselves near the chloroplasts, mitochondria act as buffers, absorbing excess energy and neutralizing ROS, ensuring that the cell continues to function even under stress. This intimate organelle embrace feels almost protective, like one partner shielding the other from harm in a moment of danger. Beyond their physical proximity, these organelles communicate through a sophisticated network of chemical signals. Chloroplasts can release ROS, calcium ions, and specific metabolites that serve as messengers to the mitochondria and to the nucleus, the control center of the cell. This process, known as retrograde signaling, allows the plant to detect stress and respond at a genetic level. Within hours of extreme heat or drought, thousands of genes are activated or suppressed, producing proteins that stabilize chloroplasts, enhance mitochondrial respiration, and regulate antioxidant defenses. Some of these proteins are shared between the two organelles, performing dual roles in energy production and stress mitigation. Others, such as alternative oxidase in mitochondria, provide a unique pathway that allows energy production to continue while minimizing ROS formation, a crucial adaptation that prevents cellular damage under heat stress. It is as if the entire cell rises together in a unified emotional response, rushing to defend, protect, and repair itself. The metabolic interplay between chloroplasts and mitochondria extends further. Under challenging conditions, chloroplasts can export small molecules, such as malate and triose phosphates, to mitochondria. These molecules serve as fuel for respiration, helping mitochondria produce ATP and carbon skeletons necessary for amino acid synthesis and other vital processes. In this way, the plant coordinates photosynthesis and respiration, ensuring that energy production matches cellular demand and that excess energy does not result in oxidative stress. This partnership is particularly pronounced in plants that experience frequent heat or water stress, such as desert species, and those with C4 or CAM photosynthetic pathways. Their cellular systems have evolved to maximize energy efficiency while protecting the delicate machinery of life from environmental extremes. The emotional resilience embedded in these interactions is staggering, like a living promise that life will fight to endure. The communication between organelles also involves calcium, a versatile signaling molecule. Under stress, calcium concentrations spike transiently within chloroplasts and mitochondria. These calcium signals activate specialized proteins known as calcium-dependent protein kinases, which in turn regulate the activity of enzymes and transcription factors essential for stress response. Even the physical structure of chloroplasts adapts, forming extensions called stromules that can reach toward mitochondria, facilitating both communication and metabolite exchange. Through this remarkable coordination, the plant can fine-tune its cellular processes, balancing energy capture, protection, and growth, all within the microscopic space of a single cell. These tiny pulsations of calcium feel almost like the heartbeat of the plant, rhythmic, urgent, alive. Research has revealed that these interactions are not just fascinating, they are vital for survival. Plants with mutations in key proteins involved in organelle communication often exhibit reduced tolerance to heat, drought, and high light. They may produce excessive ROS, experience damage to photosynthetic machinery, or fail to maintain ATP production, ultimately compromising growth and reproduction. Conversely, plants with enhanced organelle crosstalk display remarkable resilience, maintaining photosynthesis and metabolic balance even under extreme stress. Proteomic and transcriptomic studies have identified hundreds of proteins and genes that participate in this network, highlighting the complexity and sophistication of these intracellular partnerships. The contrast between vulnerability and resilience evokes a deep emotional appreciation for how fragile and how strong life can be. The implications of these discoveries extend far beyond the microscopic world of the plant cell. Understanding how chloroplasts and mitochondria coordinate their functions under stress provides insights into how plants survive and adapt to challenging environments. For agriculture, this knowledge is invaluable. By selecting or engineering crops that enhance organelle interactions, scientists may be able to improve heat and drought tolerance, ensuring stable yields in regions affected by climate change. Already, experiments with rice and maize have demonstrated that overexpression of stress-responsive mitochondrial proteins can reduce damage under heat and water stress, providing a promising avenue for crop improvement. It sparks hope. hope for food security, for adaptation, for a future where life continues to thrive despite a changing planet. Moreover, these findings illuminate the remarkable ingenuity of evolution. In harsh deserts, arid steppes, and sun-baked plains, plants have developed microscopic strategies that enable survival against all odds. The dynamic interplay between chloroplasts and mitochondria is one such strategy, a testament to the complexity, adaptability, and resilience of life. Every spike of calcium, every transient contact between organelles, and every molecule exchanged tells a story of survival, coordination, and precision. Through these unseen interactions, plants maintain the balance of energy, protect themselves from damage, and continue to grow in environments that would seem hostile to life. It is a quiet miracle, life whispering to itself, "I will endure." As researchers continue to uncover the secrets of organelle communication, they reveal a blueprint for resilience, one that combines energy efficiency, stress signaling, and adaptive metabolism. These discoveries not only enhance our understanding of fundamental plant biology, but also provide a roadmap for designing crops and ecosystems capable of thriving in a warming, unpredictable world. In every leaf, every mesophyll cell, and every organelle, there lies a story of survival, a quiet, elegant testament to the power of coordination and adaptation that allows life to endure even under the most extreme conditions. It is a story that stirs the heart, a reminder that even in silence, even at microscopic scales, life fights fiercely, beautifully, and bravely. Leaf morphological adaptation in thermophilic plants in deserts, semi-arid plains, and sun-scorched mountains, leaves tell a story of survival. Unlike in temperate forests, where broad, lush leaves soak up sunlight with abandon, the leaves of thermophilic plants, the species that thrive in extreme heat, are masters of restraint, efficiency, and protection. They are sculpted not just by chance, but by millions of years of evolution, shaped to capture sunlight without overheating, to photosynthesize without wasting precious water, and to withstand conditions that would desiccate ordinary plants within hours. In these environments, every curve, every thickness, every microscopic hair on the leaf surface serves a purpose. It feels as though each leaf carries the emotional weight of its environment, a living testament to courage and adaptation. Consider the leaves of the creosote bush, Larea tridentata, which dominate the North American deserts. From a distance, they might appear small and unremarkable, but under close inspection, they reveal an extraordinary design. Narrow and elongated, these leaves reduce the surface area exposed to the sun, while promoting airflow that cools their surface, dissipating heat efficiently. In contrast, succulent species such as Agave americana store water directly within their thick, fleshy leaves, creating a buffer against the daily assault of sun and wind. Each type of leaf represents a delicate balance between capturing energy and conserving resources, demonstrating the intimate connection between form and function in plant survival. There is a quiet beauty in this balance, an emotional harmony between survival and simplicity. Leaf orientation adds another layer of sophistication. Many desert plants engage in what scientists call paraheliotropic movements, adjusting the angle of their leaves throughout the day. During the hottest hours, leaves rotate or tilt to minimize exposure to direct sunlight, preventing overheating and reducing water loss through evaporation. In Pistacia lentiscus, for example, leaves twist along their axes, casting themselves in shadows during midday, then reopen as the sun sinks lower in the sky. This dynamic behavior is controlled by a combination of light-sensitive receptors and circadian rhythms, revealing a complex interplay between environmental cues and internal biological clocks. It is almost emotional to witness, a leaf dancing with the sun, bending but never breaking. At the microscopic level, leaf surfaces are marvels of adaptation. A thick, waxy cuticle often covers the epidermis, reflecting sunlight and reducing water loss. In Opuntia ficus indica, tiny crystals of wax scatter light, lowering leaf temperature by several degrees even under intense desert sun. Trichomes, or microscopic hairs, further enhance survival. Some form dense mats that trap humid air, slowing transpiration, while others are glandular, secreting compounds that deter herbivores and protect against harmful ultraviolet radiation. These seemingly simple structures are, in fact, intricate solutions honed over eons to preserve life in an unforgiving landscape. One cannot help but feel admiration. These tiny structures embody resilience in its purest form. Beneath the surface, the internal structure of leaves contributes equally to survival. The palisade mesophyll, responsible for capturing light and performing photosynthesis, is often elongated and densely packed, maximizing energy absorption without creating hotspots that could damage chloroplasts. The spongy mesophyll allows for efficient diffusion of carbon dioxide, even when stomata, the tiny pores on leaf surfaces, are partially closed to conserve water. In many desert plants, specialized water-storing tissue called hydranchima cushions the photosynthetic cells from dehydration, providing both moisture and thermal insulation. High-resolution imaging of Crassula perfoliata reveals a compartmentalized system of hydranchima, demonstrating how structural innovation at the cellular level supports survival under extreme heat. This hidden architecture evokes a deep emotional respect, an inner fortress built for endurance. Stomatal distribution and behavior further exemplify the precision of adaptation. In thermophilic plants, stomata are often recessed into the leaf surface or concentrated on the lower side of the leaf, reducing exposure to sunlight and slowing water loss. During peak heat, stomata close, conserving water while limiting carbon dioxide intake. When temperatures drop in the evening or after a rare rain, they reopen, allowing photosynthesis to resume efficiently. This remarkable responsiveness is orchestrated by plant hormones, such as abscisic acid, which signals cells to adjust stomatal aperture in response to environmental stress. In species like Zygophyllum xanthoxylum, genetic studies have shown that transcription factors activated by heat and drought coordinate this process, turning on or off hundreds of genes that fine-tune leaf physiology to the environment. It feels like witnessing a delicate emotional intelligence, leaves responding intuitively to the world around them. Vein architecture, too, plays a critical role. Leaves with high vein density can distribute water more efficiently, ensuring that photosynthetic cells remain hydrated even when external conditions are harsh. This redundancy allows the plant to continue growing even if some veins become blocked or damaged by heat-induced embolism. In Nerium oleander, the tightly spaced minor veins rapidly redistribute water, maintaining leaf turgor and enabling photosynthesis under intense sunlight. This internal plumbing, invisible to the casual observer, is essential to the survival of plants in extreme habitats. There is something profoundly moving about this unseen perseverance, an internal strength that never reveals its struggle. The evolutionary story behind these adaptations is equally remarkable. Across deserts, mountains, and other arid regions, unrelated plant species often display strikingly similar leaf traits, a phenomenon scientists call convergent evolution. Narrow leaves, thick cuticles, sunken stomata, reflective surfaces, and water-storing tissues have evolved independently in cacti, agaves, creosote bushes, and many other plants. Despite their different ancestries, these plants have arrived at comparable solutions to the same set of environmental challenges, underscoring the power of natural selection in shaping form to function. It is an emotional reminder that even across vast distances and histories, life discovers similar paths to survival. From a human perspective, these leaf adaptations offer both inspiration and opportunity. By studying how thermophilic plants manage sunlight, water, and heat, scientists are developing strategies to improve crop resilience in the face of climate change. Selecting or engineering crops with modified leaf thickness, trichome density, or stomatal behavior could enhance water use efficiency, reduce heat stress, and maintain photosynthesis under conditions that would otherwise diminish yields. Moreover, understanding leaf energy balance helps guide precision agriculture practices, ensuring optimal growth even in semi-arid or high-temperature regions. There is hope here, a powerful emotional hope that nature's wisdom can guide humanity through uncertain times. Leaves, often taken for granted in everyday observation, are in fact living marvels of engineering. Each curve, tilt, wax coating, and hair is a product of millions of years of evolutionary fine-tuning, designed to balance the need for energy capture with the necessity of survival. In the desert, where the sun beats down without mercy and water is scarce, these adaptations are the difference between life and death. They reveal not only the resilience of plants, but also the elegance of natural design, a design that continues to inspire scientists, farmers, and anyone who stops to study the quiet, green survivors of the world's hottest places. Their silence carries emotion, the quiet pride of endurance. Ultimately, the story of leaf adaptation in thermophilic plants is a story of balance, ingenuity, and endurance. Every structural feature, from the microscopic wax crystal to the orientation of the entire leaf, serves a purpose, orchestrating a delicate equilibrium between light, heat, water, and life itself. By exploring these adaptations, we glimpse the ingenuity of evolution and the profound ways in which life can persist, even under the most extreme conditions. These leaves are not just green surfaces. They are testament to survival, resilience, and the enduring power of nature's design. They feel like tiny green warriors, soft in appearance, fierce in spirit. Transcriptomic reprogramming in heat stress. In the hidden world within every plant, a quiet and determined revolution takes place when the temperature rises. Unlike animals, plants cannot move to escape the heat. They stand rooted in place, facing the intensity of the sun with a kind of silent courage. Instead of fleeing, they rely on a remarkable inner strength, the ability to reprogram themselves at the molecular level, adjusting thousands of genes to survive and even thrive under stress. This process, known as transcriptomic reprogramming, is one of the most intricate and awe-inspiring strategies plants use to cope with extreme heat. By changing which genes are expressed and when, plants orchestrate a delicate symphony of protective mechanisms that safeguard their growth, metabolism, and reproduction, even in the most unforgiving environments. When heat stress strikes, the first responders are a set of genes called heat shock proteins, or HSPs. These proteins act like compassionate molecular chaperones, rushing to stabilize other proteins that would otherwise denature and lose function under high temperatures. In desert-adapted species such as Tidstromia oblongifolia and Agave americana, heat shock proteins are produced in extraordinary quantities within hours of exposure to intense sunlight. These chaperones gently bind to critical enzymes in the chloroplasts and mitochondria, preventing them from unfolding and aggregating, a potential cellular disaster that could halt photosynthesis and energy production. The speed of this response is astonishing and deeply moving. Within just a few hours, the plant has already begun protecting its most vital molecular machinery, fighting quietly but fiercely for survival. But heat shock proteins are only the beginning of the plant's emotional, internal story. Transcriptomic studies have revealed that thousands of genes undergo coordinated changes in expression during heat stress. Some genes rise powerfully to ramp up the production of antioxidants, molecules that neutralize harmful reactive oxygen species generated by stressed chloroplasts. Others produce osmoprotectants, small but life-saving molecules that help cells maintain turgor and prevent dehydration as precious water evaporates from the leaf surface. Still others work to control membrane fluidity, ensuring that the delicate lipid layers of cells remain intact even when temperatures soar. This multi-layered response is anything but random. It reflects a gently refined and highly evolved network of regulatory pathways, a system of inner wisdom that works together to maintain balance during extreme heat. Central to this reprogramming is the plant hormone abscisic acid, or ABA. When heat or drought touches a plant, ABA levels rise rapidly, like an urgent message carried through its veins. This signaling molecule tells stomata to close, reducing water loss and activates stress-responsive genes. Transcription factors, the plant's molecular decision makers, respond to these ABA signals, turning on protective pathways while temporarily slowing non-essential processes such as growth. In species like Zygophyllum xanthoxylum, researchers have identified transcription factors such as DREB2A and AREB, one that act like master switches, coordinating hundreds of genes that collectively enhance heat tolerance. These transcription factors are the quiet leaders of the plant's internal world, ensuring that it can endure hardship without sacrificing long-term survival. Modern genomic technologies have allowed scientists to observe this transcriptomic reprogramming in unprecedented detail. Using RNA sequencing, researchers can map exactly which genes are switched on or off under heat stress. In studies of Arabidopsis thaliana, a model plant species, heat stress triggers the upregulation of genes involved in protein folding, antioxidant defense, and carbohydrate metabolism, while downregulating genes associated with cell division and growth. This selective prioritization is a touching reminder of how plants choose survival over expansion when pushed to their limits. Similar patterns have been observed in desert and tropical plants, suggesting that transcriptomic reprogramming is a universal strategy for coping with extreme heat, a shared language of resilience across the plant kingdom. Transcriptomic reprogramming extends far beyond a single organ or tissue. Leaves, roots, and stems each respond differently, reflecting the unique roles they play in survival. Leaves, the primary sites of photosynthesis, focus on producing protective proteins, antioxidants, and osmoprotectants. Roots concentrate on water uptake and hormone signaling, acting as the plant's emotional and physical foundation. Stems serve as connectors, integrating signals and coordinating the flow of water, nutrients, and hormones throughout the entire organism. This systemic response highlights the profound unity within a plant, every part working together, harmonizing in the face of stress. The speed and flexibility of transcriptomic reprogramming are essential for survival. In nature, heat waves can sweep through in a matter of hours or days, and plants must adapt immediately. In some species, genes are pre-primed, ready to activate at a moment's notice. In others, rapid signaling pathways allow new protective genes to be expressed within minutes. Calcium ions, reactive oxygen species, and other secondary messengers act like molecular alarm bells, initiating swift changes in gene expression. The result is a plant capable of responding dynamically to ever-shifting conditions, maintaining photosynthesis, water balance, and growth, even when the sun becomes harsh and overwhelming. Understanding transcriptomic reprogramming has profound implications for agriculture and climate adaptation. By identifying the genes and regulatory networks that confer heat tolerance, scientists hope to develop crops capable of withstanding the frequent and severe heat waves of our warming world. Genetic engineering or selective breeding could enhance the expression of heat shock proteins, antioxidants, and transcription factors in crops such as wheat, rice, and maize, helping them remain resilient without compromising yield. Beyond food crops, this knowledge can support the cultivation of bioenergy plants and sustainable landscaping in climate-affected regions, nurturing ecosystems that can thrive in adversity. Transcriptomic studies also reveal the elegant balance plants maintain between survival and growth. When protective genes are activated, growth-related genes step back, allowing the plant to prioritize life over expansion. But as soon as the heat subsides, the transcriptional program shifts once more, permitting growth, reproduction, and development to resume. This dynamic, almost poetic flexibility highlights the quiet sophistication of plant survival strategies, an inner resilience not of brute strength, but of precise, thoughtful adaptation. Ultimately, the story of transcriptomic reprogramming is a story of adaptation, precision, and profound resilience. Each plant cell acts as a miniature command center, constantly monitoring its environment, interpreting signals, and executing complex genetic programs. Through these processes, plants can survive, thrive, and reproduce in conditions that would overwhelm less adapted organisms. The invisible dance of genes and proteins, the rapid orchestration of stress responses, and the delicate balancing of energy, water, and growth reveal a world of biological brilliance often hidden from sight. In every leaf, root, and stem, transcriptomic reprogramming empowers life to endure, a quiet but powerful testament to nature's ingenuity and the enduring strength of plants in the face of extreme heat. Photosynthetic efficiency and thermal dissipation. On a sun-baked afternoon in the desert, when the ground shimmers with heat and the air itself seems to tremble. Temperatures soar to levels that would wither most forms of life. Yet remarkably, many plant species stand unfazed, continuing the miraculous, life-sustaining process of photosynthesis. At the heart of this quiet perseverance lies a delicate balance, capturing precious light energy to produce sugars, while also protecting themselves from the scorching intensity that threatens to overwhelm them. In these extreme environments, plants have evolved extraordinary mechanisms to maintain photosynthetic efficiency while gently dissipating thermal energy, a graceful combination of strategies that allows them not only to survive, but to flourish where survival seems nearly impossible. The challenge they face is immense. Sunlight is both a blessing and a danger. It fuels photosynthesis, but when its intensity surpasses what chloroplasts can safely handle, the excess energy sparks the formation of reactive oxygen species, highly damaging molecules capable of harming DNA, proteins, and cell membranes. Plants must walk a fragile line, harnessing the power of sunlight while preventing the destructive forces hidden within it. This continuous balancing act is central to their survival in deserts, high-altitude mountains, and tropical regions where heat and light press upon them relentlessly. One of the most beautiful and effective strategies they use is non-photochemical quenching, or NPQ. This elegant process allows chloroplasts to safely convert excess light energy into heat, releasing it harmlessly back into the environment. Specialized proteins, including the PSBS protein and components of the xanthophyll cycle, serve as vigilant guardians, sensing when chlorophyll molecules become overexcited. In many desert plants, NPQ activates within minutes under blinding sunlight and relaxes just as quickly when conditions soften. This quick shift reveals a plant's astonishing ability to protect itself, responding like a living, breathing system, adjusting to every beam of light. Through NPQ, plants show a kind of quiet wisdom, sustaining efficiency while preventing cellular damage. Leaf morphology, too, contributes profoundly to thermal control. The very shape and posture of a leaf can influence how much light it captures and how efficiently it cools itself. Narrow, elongated leaves reduce direct exposure, allowing air to flow more freely and cool the surface. In species such as Lyria tridentata and Agave americana, thick leaves hold multiple layers of palisade and spongy mesophyll, distributing absorbed light and heat more evenly and preventing dangerous hotspots. Some plants even reposition their leaves to avoid the harshest sun of midday, a tender, almost thoughtful gesture known as paraheliotropism. Through these adaptations, plants combine structure and biochemistry with almost artistic precision to maintain the delicate dance between energy capture and heat release. Inside the cells, chloroplast movements add another layer of resilience. Chloroplasts are surprisingly dynamic, shifting positions depending on light intensity. Under harsh light, they retreat to the sides of the cell to reduce exposure. Under gentle light, they gather where they can capture as much energy as possible. High-resolution imaging of Arabidopsis thaliana and desert species reveals that these movements occur astonishingly fast, often within minutes, highlighting a plant's ability to adapt almost instantaneously to shifting environmental demands. It is as if each chloroplast is an intelligent traveler, seeking the safest and most productive place to be. Specialized leaf structures offer further protection. Epicuticular waxes, sparkling like tiny crystals, reflect a portion of incoming sunlight, reducing heat absorption. Trichomes, tiny hairs, create a soft, insulating layer of humid air over the leaf, slowing water loss while reflecting excess radiation. Some species even produce reflective pigments such as anthocyanins and carotenoids, which act like shields against photooxidative stress. Together, these features form a resilient microenvironment, allowing photosynthetic enzymes to continue functioning even when the world around them burns with heat. Beneath these physical adaptations lies a remarkable molecular intelligence. Plants coordinate photosynthesis and heat dissipation through intricate networks of regulatory pathways. Heat stress triggers sweeping changes in gene expression that strengthen protective defenses, including antioxidants, heat shock proteins, and enzymes of the xanthophyll cycle. Calcium signaling and reactive oxygen species act as internal messengers, translating environmental shifts into carefully orchestrated cellular responses. This integration ensures that, even when the world shifts abruptly, plants can maintain both energy production and cellular safety. Stomatal regulation adds yet another layer of adaptability. The tiny pores on leaf surfaces open and close with exquisite timing, managing water loss and cooling. Transpiration acts like a plant's version of sweating, releasing water that cools the leaf. Yet in the desert, where water is precious, plants often close their stomata during the hottest hours, relying instead on their other adaptations to prevent overheating. When temperatures ease, the stomata reopen, resuming both cooling and gas exchange. Through this choreography, plants demonstrate a profound ability to balance water conservation with temperature control. Photosynthetic efficiency and thermal dissipation do not function in isolation. They are part of a deep, interconnected system that spans chloroplast-mitochondria interactions, transcriptomic reprogramming, and leaf morphology. In the desert, a single leaf may reflect sunlight with wax crystals, dissipate heat through NPQ, reposition chloroplasts within minutes, and tightly regulate stomatal openings, all simultaneously. This orchestration of strategies showcases the elegance and complexity of plant survival, particularly in environments where the sun's intensity would instantly halt photosynthesis in less adapted species. Studying these processes holds great promise for agriculture and climate resilience. By understanding how plants maintain photosynthetic efficiency under heat, scientists can shape crops capable of withstanding rapidly warming climates. Improvements may include enhanced NPQ capacity, more reflective leaf surfaces, optimized chloroplast movement, or smarter stomatal control. Such innovations could safeguard food production across regions increasingly threatened by heat waves. Beyond scientific advancement, the study of photosynthetic efficiency and thermal dissipation invites us into a deeper appreciation for the resilience of life. Plants, though rooted and unable to escape their surroundings, have evolved brilliantly adaptive systems, morphological, biochemical, and molecular, that allow them not merely to endure but to flourish. Each leaf, each cell, each chloroplast contributes to this delicate balance, revealing the astonishing adaptability of life in the face of heat and hardship. Ultimately, these strategies are more than survival mechanisms. They are expressions of nature's quiet intelligence. Every reflective wax crystal, every rapid gene activation, every shifting chloroplast reflects a solution refined across millions of years. Together, they remind us of the subtle beauty of survival, the silent choreography of cellular life, and the enduring resilience of plants beneath the sun's most relentless rays. Water use efficiency and thermal adaptation. In the arid silence of deserts and sun-scorched plains, where the earth cracks with thirst and the air shimmers with heat, plants carry out one of the quietest, most extraordinary acts of survival on our planet. In these unforgiving landscapes, where water is treasured like a sacred gift, every leaf and every root must make delicate choices. How much water to keep, how much to give, and how to stay alive beneath a sky that shows no mercy. At the heart of their survival lies a fragile balance, the dance between gathering carbon dioxide for photosynthesis and conserving the precious water that sustains life. This balance, known as water use efficiency, becomes the difference between thriving and withering. Desert plants like the creosote bush, the agave, and the gentle, patient cacti show us just how brilliant nature can be. Their leaves often glisten with protective waxes, their stomata sink deep within tiny pockets of shade, and their surfaces reflect the burning sunlight like small mirrors of resilience. Under the guidance of the hormone ABA, their stomata open and close with the rhythm of the day, a rhythm shaped by survival, by intuition, by millions of years of quiet adaptation. Inside each leaf, the tissues are arranged with breathtaking precision. The palisade and spongy mesophyll sit close together, shortening the path that carbon dioxide must travel, allowing photosynthesis to continue even when the stomata can only dare to open a little. Many plants store water in hidden tissues, small reservoirs tucked gently within their leaves, reservoirs that cushion them against dehydration and shield them from the heat. These reservoirs do more than store life, they protect it. Beneath the soil, another story unfolds. Deep taproots stretch downward like whispered prayers, searching for hidden water far below the surface. Fine lateral roots spread outward, ready to drink the rare moments of rainfall before the desert heat pulls it away. Together, deep and shallow, patient and swift, they create a network of hope beneath the sand. And then there is CAM photosynthesis, one of nature's most elegant solutions. In the cool hush of night, while the desert rests, CAM plants gently open their stomata and breathe in the carbon dioxide they need. They hold it safely as malic acid until the morning, when the sun rises and the heat returns. Then, with their stomata closed tight, they quietly carry out photosynthesis, using the carbon they stored while sparing every drop of water. A quiet miracle happening every night in silence, in darkness, in perfect adaptation. To keep themselves cool, these plants cloak their surfaces in waxes, hairs, and pigments that shimmer softly under the sun. Some tilt their leaves away from the brightest rays, like someone turning their face from a harsh light. Even the colors they produce, the soft reds of anthocyanins, the warm golds of carotenoids, help shield their inner workings from harm. Deep within their cells, a symphony of molecular signals guides them through stress. Calcium pulses, reactive oxygen whispers, transcription factors awaken, weaving together a tapestry of responses that protect the plant from heat and drought. In species like Zygophyllum xanthoxylum, master regulators work tirelessly, ensuring stomata behave wisely, osmolytes gather to hold water, and proteins remain stable in the heat. Every molecule plays its part, every signal carries meaning. Every reaction holds life steady against the world outside. These adaptations do more than protect individual plants, they shape entire landscapes. Plants that master water use efficiency become anchors of survival, forming the living framework of desert ecosystems. Around them, insects, mammals, and microbes find shelter, nourishment, and balance. Through their quiet resilience, they support life far beyond themselves. And as our world warms and drought touches more and more lands, the lessons of these desert survivors become a gentle guide for our future. Their strategies, CAM photosynthesis, precise stomatal control, reflective leaf surfaces, molecular resilience, may become the key to feeding humanity in the decades ahead. In the end, water use efficiency and thermal adaptation are not just scientific terms. They are stories of endurance, patience, and quiet brilliance. They are reminders that even in the harshest corners of Earth, life finds a way gracefully, intelligently, and beautifully. Every leaf, every root, every shimmering grain of wax speaks of the profound resilience woven into the fabric of nature, a testament to how life survives, even under the sun's most relentless gaze. Root adaptations and hydraulic conductivity beneath the blistering sun, under soils cracked by heat and time, lies a quiet, unseen world where life persists with remarkable grace. Here, in the cool darkness below the surface, roots stretch, search, and survive, forming the hidden lifelines of plants that endure some of the hottest, driest places on Earth. While leaves drink sunlight and turn it into energy, it is the roots that anchor the plant to life itself, connecting it to water, to nutrients, to stability. Their story is silent, but extraordinary. Roots must solve a delicate paradox. They need to explore vast stretches of soil, searching for droplets of water in a land that rarely gives. Yet they must not waste precious energy on empty, barren ground. Desert plants answer this challenge with a breathtaking strategy: deep, powerful taproots that descend like anchors into the earth, and wide, delicate lateral roots that spread across the upper soil like open hands waiting for rain. In plants like mesquite and Ziziphus spina Christi, these roots shift and grow with the moods of the environment. During droughts, the taproot pushes deeper, following the whisper of underground moisture. After rainfall, the shallow roots awaken, spreading outward to catch every fleeting drop before the sun steals it away. This elegant dual strategy allows the plant to survive day by day, season by season, even in worlds where water arrives only in rare, gentle moments. Inside each root, a microscopic masterpiece unfolds. Narrow xylem vessels, tiny channels carved by evolution, carry water upward, drop by drop, to the thirsty leaves above. Their strength and narrowness protect against cavitation, the dangerous formation of air bubbles that can break the flow of water and threaten the plant's survival. In species like Caligonum comosum, these vessels are reinforced like natural steel, resisting collapse under extreme tension from dry soils. Aquaporins, delicate protein gateways, open and close with perfect timing, guiding water softly into the root cells. Under drought, these gates adjust themselves, balancing water conservation with the need to sustain life. Each adjustment, each molecular shift reflects a stunning precision, the kind only nature could design. But roots do not face the desert alone. They form gentle alliances with soil fungi, mycorrhizae, whose thread-like hyphae reach far beyond the plant's own reach. Together, root and fungus share a deep reciprocity. The plant gives sugars, and in return receives water and nutrients drawn from the farthest corners of the soil. In the harshest ecosystems, These partnerships are not just helpful, they are the difference between living and fading away. Hydraulic conductivity, the plant's ability to move water upward, is a dynamic dance, constantly shifting with the world outside. When soil is moist, roots drink freely, sending water upward to cool the leaves through transpiration. But when drought arrives, conductivity slows, stomata tighten, and the plant enters a quiet, protective state. Root hairs expand the root's touch, absorbing water from the tiniest pores of the soil. Some species grow protective layers or air-filled spaces that help store and transport water efficiently, guarding the plant from heat and dehydration. Deep within this hidden world, hormonal signals guide every decision. ABA and auxins steer root growth toward the promise of moisture. Calcium messages travel from root tips to leaves, coordinating stomatal behavior and growth. The entire plant listens to the roots, adjusting its physiology to match the conditions below. A quiet, constant conversation that keeps life in balance. Roots also offer protection from heat itself. By carrying water upward, they help cool the leaves, preventing the delicate machinery of photosynthesis from overheating. Some roots reach cooler underground layers, buffering the plant against sudden spikes of desert heat. In species like Acacia tortilis, deep taproots tap hidden aquifers, secret stores of life beneath the ground, sustaining photosynthesis, even under the fiercest sun. Throughout deserts, savannas, and dry plains, evolution has sculpted these root systems with remarkable consistency. Unrelated species develop the same deep taproots, the same lateral networks, the same strengthened xylem, a testament to the relentless pressure of water scarcity. These adaptations don't just keep individual plants alive, they shape whole ecosystems. Plants with strong root systems anchor the soil, create microhabitats, and support entire webs of life. Today, the secrets of these desert roots guide modern agriculture. By understanding how plants manage water transport under extreme stress, scientists are developing crops that can thrive in a warming, drying world. Enhanced xylem strength, improved aquaporin regulation, drought-ready root architecture. These lessons from the desert may help feed the future. In the end, root adaptations and hydraulic conductivity reveal a deep and quiet truth. Even in the harshest places, life finds a way. It digs deep, reaches far, and builds partnerships below the surface where no one can see. Through intricate designs and patient resilience, roots uphold the entire plant, unseen, uncelebrated, yet essential. With every taproot descending into the earth, every lateral root stretching toward a brief rain, every microscopic channel carrying water upward, we witness the hidden elegance of survival. A reminder that beneath the world we see lies a vast, silent network of hope, endurance, and extraordinary design. Stomatal regulation and gas exchange. In the silent ballet of plant life, stomata, those tiny, unassuming pores scattered across each leaf, step forward as quiet dancers in nature's most delicate performance. Invisible to the casual observer, they choreograph a breathtaking balance, taking in the carbon dioxide plants need to live, while guarding every precious drop of water. This isn't just a mechanical act. It is a deeply intuitive, dynamic response, a moment-to-moment conversation between the plant and the world around it. Through these tiny openings, plants breathe, survive, and adapt, even in places where the sun blazes. The air cracks with heat, and water is a rare and treasured gift. The challenge is simple to describe, yet profound in its consequences. To open the stomata is to welcome life-giving carbon dioxide, but also to risk losing life-sustaining water. In the desert, where each molecule of moisture can mean survival, this decision becomes a quiet struggle for existence. Plants like Zygophyllum xanthoxylum, Laurea tridentata, and the sturdy desert cacti have evolved extraordinary ways to make peace with this tension. Their leaves hold fewer stomata, their pores hide in sheltered pits or grooves, or stay on the cooler underside of leaves, safe from the scorching gaze of the sun. Some of the most remarkable survivors, the cam plants, even learn to breathe at night. While the world sleeps, they open their stomata under the cool, dark sky, drawing in carbon dioxide when the air is gentle and moist, conserving every drop of water with quiet wisdom. Inside each stoma, a world of signaling and communication comes alive. The hormone abscisic acid, ABA, acts like a soft, urgent whisper when the plant senses thirst or heat. "Close the gates," it signals. And the guard cells obey, shifting ions, releasing water, and folding inward until the pore gently seals. When relief returns, when the soil is kinder, the air cooler, ABA retreats, water flows back, the guard cells swell again, and the stomata reopen, ready to greet the world. Other messengers join this inner conversation. Little pulses of calcium, flickers of reactive oxygen species, each one carrying signals of light, temperature, and drying soil. Together, they guide the stomata with exquisite timing, allowing the plant to breathe without breaking its fragile water balance. Over longer seasons, plants reshape themselves in response to struggle and stress. Seedlings in drought grow with fewer, smaller stomata, tiny choices that help them survive their earliest, most vulnerable days. Plants exposed to relentless heat or light gradually fine-tune their guard cells, becoming wiser, more resilient with time. Through the stomata, even the whole body of the plant is affected. Transpiration cools the leaves, pulls water upward from the roots, carries nutrients to every cell. Too much water lost and danger approaches. Too little transpiration and the leaf overheats. So desert plants weave a beautiful harmony, reflective surfaces, tiny hairs, and clever leaf shapes, helping the stomata maintain that perfect balance of breath and water. Modern science has illuminated the genetic script behind all of this. Genes like DREB, AREB, and speechless help shape how many stomata form, how they respond, and how efficiently a plant uses water. By understanding these genes, scientists are shaping the future of agriculture, helping crops like wheat and maize thrive in a warming, drying world. Beyond individual plants, these adaptations ripple outward into entire ecosystems. Plants that master stomatal regulation anchor deserts, feed communities of animals, and stabilize soils. Their success becomes the foundation on which countless other lives depend. Across continents and climates, nature repeats these patterns: recessed stomata, nighttime opening, reduced density, whispering the same ancient truth. Survival is not brute endurance. It is thoughtful regulation, quiet intelligence, and a graceful surrender to the rhythms of the environment. In the end, each opening and closing of a stoma is a small miracle, a moment in which the plant balances life and loss, breath and water, hope and survival. Through this microscopic gateway, plants endure blistering heat, long droughts, and intense sunlight, maintaining the photosynthesis that powers their growth and sustains the world around them. In the gentle workings of these tiny pores, we glimpse the deep resilience and elegant wisdom of plant life, a reminder that even the smallest openings can hold extraordinary strength. Heat shock proteins and cellular protection. In the quiet, hidden world inside every plant cell, a remarkable defense awakens the moment heat begins to rise. These gentle protectors, known as heat shock proteins or HSPs, act like tiny guardians, silent, swift, and endlessly loyal. Though we cannot see them, they stand at the front lines of survival, shielding the delicate machinery of life when temperatures climb to dangerous heights. To understand HSPs is to witness one of nature's most beautiful truths: that even in the face of overwhelming heat, life finds a way to protect itself with astonishing grace. The story of heat shock proteins began in the 1960s, when scientists first noticed a sudden burst of unique proteins appearing in fruit flies exposed to high temperatures. Soon, we learned something even more extraordinary. These guardians exist in plants across the world, from towering trees in cool forests to the brave cacti enduring desert suns. And when heat strikes, plants do not hesitate. Within minutes, their cells begin producing HSPs, as if the entire organism whispers, "Protect everything, hold the structure together, survive." One of the most vital roles of HSPs is that of a molecular chaperone, a comforting guide in a moment of crisis. Heat can twist and unravel proteins, robbing them of their shape, their function, their very identity. Left unprotected, they clump together, forming harmful tangles that threaten the life of the cell. But HSPs gently cradle these destabilized proteins, preventing them from collapsing into chaos. They help them refold, recover, and return to their essential duties. In plants like Agave americana and Zygophyllum xanthoxylum, HSPs even safeguard the precious photosynthetic enzymes, ensuring that the plant can continue capturing sunlight, even when the world around it feels scorching and relentless. Different HSP families carry out their own special missions. HSP 70 tends to newly formed or partially unfolded proteins, guiding them toward their proper shape like a patient mentor. HSP 90 stabilizes signaling proteins and transcription factors, helping stress response pathways stay steady and strong. And the small HSPs, quiet, nimble, and everywhere, rush in first, preventing protein aggregation and supporting enzymes in moments of intense heat. Together, they weave a multi-layered shield around the cell, holding everything together when it matters most. But this defense is not random. It is orchestrated with delicate precision. When temperatures climb, heat shock transcription factors, HSFs, awaken and bind to specific DNA sequences, flipping on the genes that create HSPs. Like an alarm that instantly mobilizes the entire protective system, this response allows plants to react within minutes, to defend themselves before damage takes hold. In desert plants, this genetic surge is even more dramatic. Their cells respond to heat waves with powerful waves of HSP production, often accompanied by antioxidants and osmoprotectants. Together, these molecules form a beautifully coordinated network, each one supporting the others. A symphony of survival playing out in silence. HSPs protect not only proteins, but also the fragile architecture of cellular membranes. Heat softens these membranes, making them unstable and leaky. HSPs reinforce them, like tiny hands supporting fragile walls, ensuring that ions stay balanced, that compartments stay intact, that life continues smoothly inside the cell. In chloroplasts, they protect the photosystems from overheating. In mitochondria, they keep energy production alive. Through all of this, HSPs help the plant hold onto the delicate balance that makes life possible. Their importance extends far beyond the individual plant. Species with strong HSP responses withstand heat waves, drought, and temperature swings with a quiet strength. They grow where others wither. They reproduce where others fail. And in doing so, they shape entire ecosystems, supporting animals, insects, and soil communities that depend on them for survival. In agriculture, HSPs offer a beacon of hope for a warming world. By enhancing these proteins in crops like rice, wheat, maize, and tomato, scientists have created plants that stay productive even during extreme heat. Fields remain green, yields remain stable, and humanity gains a powerful ally in the fight against climate stress. Heat shock proteins do not work alone. They partner with antioxidants, osmoprotectants like proline, and signaling molecules like reactive oxygen species. Together, they form an intricate web of protection, each molecule sensing, responding, adjusting, and reinforcing the others. This harmony reveals the astonishing intelligence woven into plant biology, a molecular choreography refined over millions of years. Evolution has shaped the HSP system with exquisite care. Plants from hot deserts and humid tropics carry larger families of HSP genes, stronger baseline protection, and faster reactions. These adaptations give them the resilience to endure the harshest of environments, proof that life not only survives challenges, it grows more extraordinary because of them. Ultimately, heat shock proteins are symbols of resilience, guardians of the microscopic world within every leaf. They protect photosynthesis, energy production, and growth. They shield cells from collapse. They hold the plant together through the fiercest heat. In the quiet, unseen depths of the cell, HSPs work tirelessly, a testament to life's unwavering determination to endure. In their silent service, we glimpse the remarkable strength and delicate beauty that allow plants to thrive on a warming earth. Antioxidant mechanisms and reactive oxygen species management. In the quiet, hidden chambers of a plant cell, sunlight is both a gentle blessing and a silent threat. It fuels life, gifting energy to every leaf. Yet when the sun grows intense, when heat lingers too long, it sparks the creation of reactive oxygen species. ROS, tiny, restless molecules capable of tearing through DNA, proteins, and membranes. Uncontrolled, they are destructive. But plants, shaped by millions of years beneath bright skies, have evolved an extraordinarily graceful defense. An intricate, intelligent network of antioxidants that protect life at its most delicate level. A system that whispers, even under scorching stress, "You are safe. Keep breathing. Keep growing." ROS form naturally as part of life's daily rhythm, during photosynthesis in the chloroplast, during respiration in the mitochondria. But when sunlight becomes too strong, when heat presses down on the leaves, electrons slip from the photosynthetic machinery and transform into superoxide radicals. In the mitochondria, stress creates hydrogen peroxide and even more reactive molecules. At low levels, ROS act as gentle messengers, signaling change and adaptation. But when they rise too high, they threaten the very heart of the cell. And so plants respond with a quiet army of protectors. Enzymatic antioxidants form the first shield. Superoxide dismutase, SOD, intercepts superoxide radicals and transforms them into safer molecules. Catalase breaks hydrogen peroxide into harmless water and oxygen. Peroxidases reach deeper still, neutralizing what remains. These enzymes work together like a coordinated rescue team, each step slowing, softening, and finally disarming the threat. In desert plants such as Zygophyllum xanthoxylum and Agave americana, these enzymes are present in remarkably high levels, even when the plant is calm, an evolutionary memory of the harsh worlds they come from. Alongside them, non-enzymatic antioxidants drift gently through the cell, offering another layer of protection. Ascorbate, glutathione, carotenoids, flavonoids. These quiet molecules act as scavengers, absorbing harmful ROS before they cause harm. Carotenoids in the chloroplasts absorb excess sunlight, releasing it as gentle heat, protecting fragile photosystems. Flavonoids in the vacuoles and outer tissues shield the plant from UV radiation and quietly neutralize ROS that slip through. Together, these compounds form a soft, glowing shield, an invisible armor wrapped around every cell. This entire defense network is beautifully coordinated. ROS themselves, though dangerous, act as signals, alerting the nucleus, activating genes, calling antioxidant enzymes into action. What could have been destruction becomes information, a warning transformed into resilience. Heat shock proteins, the guardians described earlier, work hand in hand with antioxidants. They cradle enzymes that are strained by oxidative stress, keeping them stable, functional, alive. This integration shows the deep harmony within plant biology, a system where every molecule, every signal, every response is connected in a seamless web of survival. The placement of these antioxidants is no accident. Chloroplasts hold high levels of carotenoids and ascorbate peroxidase, standing guard where light hits strongest. Mitochondria shelter their own sod and peroxidases, guarding the engines of respiration. Peroxisomes and the cytosol contribute their own defenses, ensuring no space within the cell remains unprotected. This careful positioning reflects a simple truth. Plants do not fight chaos, they organize around it. When weather changes, heat rises, drought deepens, sunlight sharpens, plants sense the shift almost instantly. Antioxidant enzymes surge in activity within hours. Non-enzymatic antioxidants accumulate like a reservoir of calm strength, preparing the plant to face repeated stress. This responsiveness allows even the most fragile seedlings to stand firm under harsh skies. ROS also play a second, gentler role, helping plants acclimate. Low levels activate pathways that increase heat shock proteins, osmoprotectants, and transcription factors. Through this dual nature, both danger and signal, ROS become part of a sophisticated language that helps plants adjust to their world. On an ecological scale, antioxidant strength becomes a silent force shaping landscapes. Plants with powerful ROS defenses endure the hottest deserts, the brightest mountain slopes, the driest plains. They anchor ecosystems, stabilize soil, and support the lives of insects, birds, and countless microorganisms. Their resilience ripples outward, affecting entire communities. And for humanity, understanding these systems brings hope. By strengthening antioxidant pathways in crops through breeding or biotechnology, we can help plants endure heat waves, drought, and intense light. Fields stay green longer, yields remain stable. Life continues in environments that test every boundary. Ultimately, the management of reactive oxygen species reveals a profound truth about plants. That survival is not just a matter of endurance, but of exquisite coordination. Through enzymes and scavengers, through compartmentalization and signaling, through integration with countless other pathways, plants transform potential harm into harmony. Every ROS neutralized, every antioxidant activated, every cell protected is a quiet triumph of evolution. A reminder that even in the harshest conditions, life does not simply resist. It adapts, evolves, and finds a way to flourish. Thermoregulation via leaf movements and orientation in the relentless heat of deserts, savannas, and sunlit grasslands, leaves become quiet warriors, brave, delicate, and determined. They are the engines that capture sunlight, yet they are also the first to face the harshness of burning days. Too much heat can harm them. But plants, in their ancient wisdom, have learned how to protect themselves through gentle movements, tiny, graceful adjustments that help them stay alive. This is the hidden ballet of thermoregulation, a dance of leaves shifting, turning, folding, and lifting in response to the world around them. It is subtle, peaceful, and deeply powerful. Leaf movements fall into three soft rhythms: heliotropism, where leaves follow the sun like curious children reaching for light; paraheliotropism, where they turn away shyly from the fiercest heat; and nyctinasty, the tender nighttime folding that feels almost like sleep. Inside each leaf base, special motor cells respond to signals of light, warmth, and hormones. They shift water, move ions, and change shape, quietly guiding the leaf to a safer angle. It is almost as if the plant is breathing, adjusting, finding comfort. In some plants, like Mimosa pudica, these movements happen in moments, a quick flutter of life. In others, the changes are slow, unfolding over long hours, a patient choreography shaped by survival. When temperatures rise, leaves tilt themselves away from the burning sun, protecting their vulnerable chloroplasts from damage. When the day cools, they open wide again, like arms welcoming the morning light. Desert plants such as Laria tridentata perform this dance every day, shifting angles, balancing light and heat with astonishing precision. But movement is only part of their armor. Narrow leaves, tiny hairs, waxy coatings, and reflective pigments all help plants manage the sun's intensity. These features shimmer lightly in the desert heat, reducing water loss and keeping the plant cool, nature's own protective clothing. By adjusting their position, leaves also save precious water. In the driest places on Earth, every drop matters. For Cam and C4 plants, where stomata open only at specific times, these leaf movements become life-saving strategies, helping them endure scorching days with quiet resilience. Deep inside the cells, heat response pathways awaken when temperatures soar. Heat shock proteins, antioxidants, and protective molecules come rushing to help. Reactive oxygen species and calcium ions send tiny signals from cell to cell, messages that guide the plant's response like whispers in the dark. These graceful adjustments don't just protect individual leaves, they shape entire ecosystems. When many plants orient their leaves together, they create cooler pockets of air, small refuges for young shoots, pollinators, and the soil life beneath them. This shared resilience makes harsh landscapes more livable for all. Understanding these strategies has become incredibly valuable for agriculture and restoration. Crop scientists now look to these natural leaf movements as inspiration, seeking heat-tolerant varieties that can thrive in a warming world. In dry regions, planting species with strong thermoregulatory behaviors can help landscapes survive and flourish again. Across evolutionary time, plants in different corners of the world have developed the same elegant solutions. With no way to walk away from danger, they learned instead to move just enough, quiet tilts, slow folds, gentle rotations to endure. This convergence speaks to the deep, universal power of adaptation. In the end, the way leaves move is more than survival. It is a testament to the intelligence of nature, an ongoing story of resilience, sensitivity, and grace. Each motion, no matter how small, reflects a delicate negotiation between life and the environment. A leaf may seem still, but in its silence, it is always listening, adjusting, and choosing its path. A quiet dance of hope beneath the sun. Osmoprotectants and cellular homeostasis, deep within the microscopic world of plant cells, life is a quiet balancing act. Every moment, tiny structures work to keep harmony within, even as the outside world becomes harsh and unforgiving. When heat scorches the air, when drought pulls the moisture away, or when salt fills the soil, the delicate chemistry of the cell begins to tremble. And yet, plants survive. They endure through a remarkable, almost tender strategy: the creation of osmoprotectants, small but powerful molecules that act like guardians, wrapping the cell in invisible protection. They are the cell's gentle shields, preserving balance, structure, and calm even during the most extreme environmental storms. These osmoprotectants come in many forms: soft, sweet sugars like trehalose and sucrose, amino acids such as proline and glycine betaine and polyols, mannitol, sorbitol, silent defenders that drift through the cell's watery spaces. Each of them serves more than one purpose. They keep proteins stable, prevent membranes from leaking, and make sure enzymes continue their life-giving work. Most importantly, they help maintain osmotic balance, ensuring the cell neither dries out nor bursts, but remains in gentle equilibrium. Proline is one of the most deeply studied of these protectors. In times of drought or heat, proline levels rise dramatically, sometimes tenfold. Like a soft cushion within the cell, it calms reactive oxygen species, stabilizes proteins, and even helps maintain the cell's internal pH. Desert plants, such as Zygophyllum xanthoxylum and Agave americana, rely heavily on this molecule, letting it gather strength inside them as temperatures climb. Proline even works hand in hand with heat shock proteins, enhancing their ability to guard the cell's machinery. It is a perfect example of the elegant teamwork within plant stress responses. Glycine betaine plays an equally beautiful role. It settles gently inside chloroplasts, protecting photosystem II like a guardian standing watch. In salty soils, habitats where life struggles to survive, halophytes build high levels of glycine betaine, allowing photosynthesis to continue, even when water is scarce. Through this molecule, plants keep energy flowing, leaves growing, and life moving forward. Sugars, too, join this quiet symphony of protection. Trehalose forms tiny hydrogen bonds that hold membranes and proteins steady during heat or dehydration, almost as if it were embracing them. Sucrose not only balances water movement, but also neutralizes harmful molecules created during stress. In plants like Opuntia ficus indica, the resilient prickly pear, high sugar levels are tied to their incredible drought survival. Polyols like mannitol and sorbitol help maintain the cell's internal water potential. By holding water gently inside the cell, they prevent dangerous dehydration and keep enzymes active in intense heat. They also act as antioxidants, calming the reactive oxygen species generated under high light or scorching temperatures. In certain desert shrubs, these polyols can make up a large portion of the cell's soluble content, evidence of their vital role in survival. Behind all of this protection lies a sophisticated web of molecular control. Transcription factors, DREB, AREB, MYB, act like sensitive messengers, turning on the genes needed to produce these osmoprotectants. When drought, heat, or salt strike, these genetic signals flare into action, increasing production exactly when it is needed. Enzymes are modified, pathways are tuned, and osmoprotectants rise like a wave of defense across the cell. These molecules do not work alone. They connect with heat shock proteins, antioxidants, carotenoids, chaperone proteins, forming an integrated shield that surrounds the cell with resilience. Proline supports antioxidant enzymes. Glycine betaine strengthens photosystem II. Sugars prevent protein aggregation. Altogether, they create harmony in the face of chaos. In nature, this ability shapes entire ecosystems. Plants that produce high levels of osmoprotectants thrive in deserts, salty plains, and heat-stricken landscapes. Their survival provides shelter and stability for pollinators, herbivores, and soil life. They become anchors of resilience, supporting biodiversity in places where life hangs by a fragile thread. And for humanity, these microscopic guardians offer hope. Scientists now harness the power of osmoprotectants to develop crops that can withstand drought, salinity, and heat, conditions that are becoming more common in a changing climate. By boosting proline or glycine betaine pathways, crops like rice, wheat, and tomato have shown greater survival and yield in harsh conditions. Osmoprotectants even help in seed priming and post-harvest storage, extending life beyond the field. Ultimately, osmoprotectants reveal the quiet ingenuity of plant life. Through these tiny molecules, sweet, simple, and profoundly effective, plants maintain homeostasis, protect vulnerable structures, and carry on their vital functions. Each molecule is a guardian, a microscopic ally, a symbol of the resilience and elegance that define the plant kingdom. Even in the harshest environments, life finds a way, softly, silently, beautifully. And now, as we drift to the end of our journey through the secret life of plants, let the quiet, green world around you wrap you in its gentle embrace. Feel the calm rustle of leaves in your mind breathe slowly, and let yourself rest in the tender rhythm of nature. If this peaceful journey touched your heart, you can softly support us by subscribing and sharing, so more souls can wander gently through the quiet wonders of life, feeling the magic in every leaf and petal. Thank you for being here, for listening, for feeling, and for pausing with us. And good night. May your dreams be as serene and vibrant as the world of plants.